Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Pushing the Boundaries of LNP Production, an innovative analytical platform for evaluating vital LNP characteristics. I'm Charlotte Barker, an editor at BioInsights, and joining me today is Nejc Pavlin, who will present a chromatographic tool that allows you to monitor key quality attributes of the LNP production process. After the presentation, we will be having a live Q&A session, so do feel free to pose your questions to Nejc using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. I'd also like to draw your attention to the Resources tab on the right, where you can find more information on the topics covered today. So now, just briefly introduce our presenter. Nejc Pavlin is a project manager in the Analytics Development Department at Sartorius BIA Separations. He has extensive experience in analytics development for a range of different biological molecules intended for therapeutic use, including pDNA, mRNA, and LNPs. So, without further ado, I'm going to hand over to Nejc to kick off today's presentation. Over to you, Nejc.
So, hello everyone. My today's talk will be about analytics of LNPs. I want to present you an innovative analytical platform developed here at Sartorius BIA Separations for evaluating vital LNP characteristics. I will start with just short introduction about our company. Then I continue with the introduction in lipid nanoparticles. I want to show you the analytical platforms for different biomolecules developed here at BIA Separation for pDNA and mRNA. And then I will turn to the main topic of today's lecture, LNP Switcher Platform. We're going to go through three different parameters of interest regarding LNPs, encapsulation efficiency, LNP size and size distribution, and lipid composition. At the end, I will perform a quick conclusion of today's webinar. So first, few words about our company. Sartorius BIA Separation is located in the center of Europe, and we are the leading developers of monolithic technology. We are exclusive producers of innovative interaction media, monolithic chromatographic columns with more than 20 years of experience, and we employ more than 200 people. We are leading experts in the purification of large biomolecules, such as different viruses, AAVs, adeno, and so on, and also for different biomolecules such as pDNA, mRNA, LNPs, and others. Here at Sartorius, we offer you solution for downstream process development, solution for manufacturing, and solution for analytical method development. Our portfolio can be divided in two different fields. First, bigger field is a preparative chromatography field. Here, our leading products are preparative columns called C-Mutus columns. This can be scalable from 1 milliliter to up to 40 liter columns. They are ready to use and are GMP compliant. Another field inside of that preparative chromatography field is screening field chromatography. Here we offer you two different solutions. One is CMYC discs. This can be modified with different chemistries to serve your personal needs. And here we also offer you SIM multi-well placed for high throughput purification and cost-effective screening of your products. The second field is analytical chromatography field. Here we have two different products. First are our CMAC analytical columns, which are designed for high resolution analysis of your biomolecules. They are specialized for different fields of biomolecules such as pDNA, AAV, etc. We offer you also the chromatographic systems called PATfix, which can be used in process control and product release quality control. This system comes with our software with preloaded and pre-validated methods, reference standards, and more. So let me start with a short introduction about lipid nanoparticles. Lipid nanoparticles are versatile and tunable drug delivery system capable of encapsulating various payloads. Their size ranging from 50 to 200 nanometers. The structure of lipid nanoparticles can be divided into two parts, I will say. The inner part of the particle where payload is encapsulated, and outer shell of the particle, which is responsible for stability of these particles. We encountered different challenges regarding LNPs. First is stability. LNP can be sensitive to temperature, to change in temperature, change in pH. They no go well with high salt concentration, and they are also sensitive to the mechanical stress. The second challenge is immunogenicity. Some LNPs may trigger an unwanted immune response in the body. And the third main challenge regarding LNPs is analytics of these particles. And there is quite a big lack of analytical methods for analysis of key parameters of lipid nanoparticles. So a little bit about synthesis and the function of lipid nanoparticles. So lipid nanoparticles are usually prepared in quite simple process with the help of microfluidic mixing of two different solutions. One solution is aqueous solution where nucleic acids are dissolved, and the other solution is ethanolic solution of different lipids. These two solutions are mixed together, and at the end we obtain the nucleic acid loaded lipid nanoparticles. When these particles are injected in the body, I would say, they get catch on the surface on the cell. Then cellular uptake of these particles is happen, and the load is released inside of the cell. Here, the translation to different proteins is happened, and these proteins then trigger the immune response of the body. So here is a typical production process of LNPs. If we want to produce LNPs encapsulating, for example, mRNA, we need to start at the beginning of this process, usually with production of pDNA. Production of pDNA is usually composed of different steps, such as purification of the lysates of E. coli cells, usually, and then the linearization of plasmid. All these steps in the production of pDNA can be nicely observed and controlled using our PATfix pDNA analytical platform, developed, I think, around 2 years ago here at BIA Separation. Then, when we have a purified and linearized plasmid DNA, this plasmid DNA can go into the IVT reaction mixture where it serves as a template for synthesis of mRNA. Also, in the production of mRNA, there's a lot of different steps, including, for example, control of IVT reaction, the control of purification process, and also all these processes can be quite nicely observed and optimized and controlled using our second platform, PATfix mRNA analytical platform developed around 1 year ago. Then when we have a purified mRNA, it goes through the encapsulation and we prepare lipid nanoparticles. Also here, a lot of parameters of interest are present. Today, I will be focusing on these three. So the encapsulation efficiency, size and size distribution, and lipid composition of the lipid nanoparticles. And all these three parameters could be controlled using our newly developed PATfix LNP analytical platform. Before I go to the LNP platform, I want to say something about our analytical platforms. So what is PATfix analytical platform? Is a chromatography platform composed of four main pillars. First one is our hardware. We use biocompatible hardware designed for analysis of biomolecules. We also have our columns, especially analytical columns with different modalities. We also have methods, analytical methods developed to work as well as possible with our hardware and our analytical columns. These methods are pre-developed and pre-validated, so you need just to use it. There is no need of any optimization, so on. And also the one important thing involving in our PATfix platform is a software. We developed software here in-house. The software is used for controlling the hardware components of the PATfix system and also serves as a platform when we can visualize the data obtained and also to work on the data obtained on the hardware. So let me say now something about our pDNA platform. pDNA platform is based on one analytical method and one analytical column, CMAC DAE column, and it's used for could be used for different reasons. So first is determination of concentration of different pDNA isoforms in any process sample during the process of preparation of pDNA. It could be used for monitoring and optimization of purification process, for lysis optimization, and for controlling plasmid linearization kinetics. Here is one example of the example chromatogram showing you the lysate of E. coli cells. This is known to be quite dirty sample, and all these components of this sample can be really nicely separated and determined using this pDNA platform. So at the beginning of chromatogram, the protein and other impurities are eluted. Then we have elution of RNA from E. coli cells. And at the end of the chromatogram, we have our analyte of interest, pDNA. It's also, with using of this method and platform, we can also separate different isoforms of pDNA, and also these isoforms can be quantified. So here is one example of our mRNA platform. Here the situation is a little bit different. We cannot analyze all the parameters in mRNA production process using just one column. So here we developed three different analytics for different purposes during the mRNA production process. For example, this platform can be used for IVT reaction monitoring and optimizing of the IVT reaction, and this is done using our CMAC Prima S column. Then we use the second column oligo-DT affinity column for quantification of mRNA during the whole production process. And also we develop a method for controlling mRNA integrity and double-strand RNA detection using our reverse phase CMAC SDVB column. Here I have one example of IVT reaction sample. So we took the sample, the aliquot of the IVT reaction mixture, and we put it on our PATfix platform, mRNA platform, and you can see that we can quite nicely separate all the components present in the IVT reaction mixture. For example, at the beginning of chromatogram, capping reagents are eluted. Here I have an example where ARCA was used. Then we have peaks representing nucleotides inside of the reaction mixture. Here is eluting pDNA template, and at the end of the chromatogram, mRNA, which is produced in the IVT reaction. So you have the information in around of your IVT reaction in around 4 minutes. So let me now go to my main part of this webinar. So the LNP Switcher platform. Here we need to change a little bit the hardware setup of our conventional PATfix system. We add additional pump, which enables us to operate this system with 2 different column modalities. The complexity of LNP sample, we cannot, due to complexity of LNP sample, we cannot use just one column to obtain the wanted parameters from the sample, so we add additional column. We also equipped with our PATfix system with switcher system with 3 different detectors. First is UV-Vis detector for detection of nucleic acid. The second one is multi-angle light scatter detector for detection of particles and for size determination. And the third detector is charge aerosol detector, which was used or is used for lipid detection. Yeah. So 2 different column modalities. We use here 2 different columns. First column in the row is our CMAC OH column, analytical column, which is used for separation of lipid nanoparticles from free nucleic acids present in the sample. It's a hydrophobic interaction column with large 6 micrometer pores. And the second column used in this system configuration is our CMAC SDVB column. We use it for separation and quantification of free and encapsulated nucleic acids. Is a reverse phase column, and we use it in the combination with ion pairing reagents. The 2 columns are connected in line and separate through valve switching, that we can select the fraction which goes from one column to another column. So here is a schematic representation of our PATfix switcher system. We can divide it in 2 parts, or we can say the 2 dimensions. So in first dimension, we use our OH column, and in the second dimension, the SDVB column is connected. The sample of LNP is usually composed of free nucleic acids and lipid nanoparticles, and we can directly inject the sample on our switcher system without any sample pretreatment. We only dilute it with the loading buffer. So when the sample is injected on the system, the lipid nanoparticles are catched on the first column, and the nucleic acids go ahead directly to the second column. When we can separate them, and analyze them using UV detector. In the next step, we elute the particles from the first column and then go through the mouse detector when we can detect it. And also this detector is used for determination of the size and size distribution of these particles. In the next step, the lipid nanoparticles travel through ahead to the second column, SDVB column, where we open it and release the load inside of the particles, and we can also obtain the individual lipids which was forming the LNP particle. Then we separate these species. Lipids are detected using a charge aerosol detector, and the nucleic acids are again determined and quantified using UVs detector. So this is a typical chromatogram obtained from LNP switcher. We can see the signals for all three detectors which were connected on the system. So first, in the upper part of the chromatogram, the UV-Vis detector is shown. Then the second, charge aerosol detector, and the third, multi-angle light scatter detector. We can divide this chromatogram in 5 different regions or parts. In the first part, the load of the sample is happened. We can really nicely control and have a control and see if everything is binded on the column, that we don't lose any species in the LNP formulation. In the second part of the chromatogram is where the free nucleic acids are detected and separated. So here is a peak representing free mRNA, for example. Then in the third part of the chromatogram, LNPs are eluted from the first column, OH column, and they can be really nicely detected using multi-angle light scatter detector. In the fourth part of the chromatogram, the analysis of encapsulated nucleic acids is present. We can see a really nice peak representing the encapsulated nucleic acids. And for the last part of the chromatogram, we analyze the lipid composition of these LNP samples. Now I want to go a little bit more in detail and show you how LNP switcher is used for determination of different parameters. So first parameter is encapsulation efficiency of encapsulation process. This is one of the, I will say, most important parameters about lipid nanoparticles. It gives us information how much mRNA is inside of the LNP particle and is protected from RNases and other stuff which can degrade mRNA, and the amount of mRNA which is present outside of the particle. Usually, ribogreen fluorescence assay is used for determination of encapsulation efficiency, and also other methods, including digestion of LNPs and determination of mRNA using spectroscopic and chromatographic methods. But here we can encounter problems, especially with extensive sample pretreatment, which can change the composition of the particles and so on. So now I want to show you the chromatograms of different samples, which was analyzed using our LNP switcher. First, I want to show you chromatograms analyzing only free nucleic acids, so without any LNP particles. So here we have a mixture of mRNA and pDNA, so 2 different nucleic acids. We can see here in UV signal that these 2 nucleic acids are really nicely separated between each other. We cannot see any peak in the mouse detector. So this is true because no particles was injected on the system, and also we have no peaks or no eluting species in the area of the chromatogram where encapsulated nucleic acids should elute. Here is another example when we analyze two different mRNAs with different sizes. So first mRNA was 1 kilobase big, and the second one was 4 kilobase long. And with using this LNP switcher, we can also distinguish between different size mRNAs potentially inside of LNP particles. And also here, no elution representing encapsulating RNA is present, and no peaks in mouse. So LNP switcher is also capable of separating mRNAs by their size. So here is the example of LNP sample encapsulating mRNA. Here, in the area of free mRNA elution, we can see the small peak representing the free mRNA inside of LNP formulation. And in the area of the encapsulated mRNA, we have also the peak representing encapsulated mRNA. With these two, we can integrate these two peaks and really nicely calculate, for example, the encapsulation efficiency of these LNP samples. We can also see the quite nice mouse response representing, shown here on the lower part of the chromatogram, where LNPs are eluting from OH column. Quite similar situation when pDNA LNP was analyzed using our LNP switcher. Nice peak representing free pDNA, nice peak representing encapsulated pDNA, and also quite nice response in the mouse detector. Here is also one interesting thing about this LNP switcher. We can also analyze the co-encapsulated LNPs. In this case, we analyze the co-encapsulated mRNA and pDNA of lipid nanoparticle, and we can see that in the area of free mRNA, two peaks are detected representing free mRNA and free pDNA, and also in the area of encapsulated nucleic acid elution, we can see both peaks presenting mRNA and pDNA. So using this system, we can also determine the amounts of individual nucleic acid free as well encapsulated in co-encapsulated LNPs. And also here in the mouse, we have quite nice response where LNPs are eluting from OH column. Okay, then we checked also the few main, I will say, parameters, validation parameters of this method. For example, we check linearity by injecting different concentration of encapsulated mRNA particles on this switcher. So here is a chromatogram presenting overlay of these the different concentrated samples. We check the linearity for free and for encapsulated mRNA, and we can see that the response is for both species is quite linear. We also check the LOD and LOQ of this method, and we can see that both parameters are around 10 nanograms of nucleic acids. We also check the recovery of encapsulation efficiency for different concentration of LNP particles. We have quite stable determination of encapsulation efficiency at different concentration of LNP particles. This was the same sample analyzed. And we also checked the recovery of free mRNA and recovery of encapsulated mRNA by spiking these two species in the null concentration of LNP particles. And we can see that we obtain a quite good result for the recovery of individual nucleic acids species. At the end, I want to show you also the comparison of LNP switcher to the results obtained using conventional ribogreen assay. So in ribogreen assay, ribogreen dye is used for detection and quantification of nucleic acids. Triton surfactant is applied for opening the LNPs. And if we compare the result obtained on LNP switcher and with ribogreen method, we can see that there is no significant difference in the results obtained with these two techniques. So I want to go ahead to the second parameter of interest, which can be determined using our PATfix system, and that is LNP size and size distribution. So usually different techniques are used for determination of size and size distribution. For example, DLS, NTA, or video drop, and also other microscopic techniques, including TEM or cryo-EM. Here are the two examples using video drop for size and size distribution determination. And also here is cryo-EM images of lipid nanoparticles from which we can also determine the size and size distribution of lipid nanoparticles. So first thing, to see if this system can be used for size and size determination, we need to check if we change somehow the sample regarding the size and size distribution when we bind it on the OH column and then also elute it from the column. So we prepare the crude sample of LNPs encapsulating mRNA, and we analyze this sample using video drop analysis. Then this sample was loaded on our OH column and eluted from it, and we collect the fraction of the LNPs eluting from the switcher system, and then analyze it again using the same technique, video drop technique. And if we see the size of the particle is almost the same as for the crude sample, we can also say that the size distribution stays quite the same when when loading and eluting from OH column is used. And one another thing which is quite interesting is that we can really nicely concentrate the particles with dilution from OH column. So some concentration is happened, but regarding size and size distribution, the sample stays the same. So here is then the example how this system can be also used for size determination of these particles. So here is the chromatogram I already showed you before of mRNA encapsulated LNPs. So here is where the elution of particles from OH column is happened. We use multi-angle light scatter detector. We measure the scattering of the light at different angles, and then by applying one calculation model, we used a Berry calculation model. we can also calculate the radius of the particles eluted from OH column. The majority of particles are eluted, and their size is around 70 nanometers, and quite the same or the same result, almost the same result is obtained when video drop was used for size determination of these same particles. So we can say that with that we can quite that these results are in quite excellent agreement with video drop measurement. Okay, and now the third parameter, which can be determined using the same technique as LNP switcher, is the lipid composition. So lipids are the main building blocks of lipid nanoparticles. We can divide it in 4 major groups of lipids, which are usually used for LNP sample preparations, LNPs preparation. So first group is phospholipids. They call it also the structural lipids, and here are some examples of phospholipids shown on the picture. The second group are ionizable or cationic lipids. Here are the 3 most commonly used lipids, cationic lipids or ionizable lipids. Then the third group are pegylated lipids, which form the surface of the particle, and also cholesterol is usually used for stabilizing the particle and also to help to transfer the LNPs inside of the cells. So here is one example how our SDVB column and increasing acetonitrile gradient can be used for separation of different lipids. The separation of these different lipids is due to the lipids are separated due to their hydrophobicity, which is related to the carbon chain length of these lipids. Using this method, we can quantify the lipids. One interesting thing is that we need to use charge aerosol detector because lipids cannot be detected with the conventional detectors, for example, UV detectors, so no peaks in the UV detector response. And this method can be really nicely adjusted and modified to separate the lipids which our clients are used for LNP preparation. So here is the example chromatogram where also charge aerosol detector was used. We can see that lipids are eluting here from 35 minutes to 41 minutes. And as I already said, this method can be easily adjusted to the desired lipid composition. So in one LNP switcher run of analyzing LNP particles, we can determine the encapsulation efficiency using UV detector. We can also determine the composition of lipids composing this particle, and with multi-angle light scatter detector, we can also determine the size and size distribution of these particles. So I'm coming to the end of today's lecture. So let me just conclude today's webinar. So LNP switcher is all-in-one analytical method designed to monitor at least 3 crucial parameters in LNP formulation: encapsulation efficiency, size and size distribution, and lipid composition in only one run. This versatile PATfix switcher is suitable for various LNPs analysis, including mRNA LNPs, pDNA LNPs, and also co-encapsulated LNPs. It eliminates the need for sample pretreatment and delivers result in less than 45 minutes. The fully customizable analytical methods for both columns allows you to tailor the analysis to your specific needs. And also when integrated with other PATfix platforms, such as pDNA platform and mRNA platform, it offers comprehensive control over the entire process from pDNA at the beginning to the final LNPs. At the end, I want to invite you to the Summer Monolith Symposium 2024. This symposium will be held in Porto Roche from June the 3rd to June the 7th next year. There will be a lot of interesting talks about nucleic acids, viruses, VLPs, and other. So yeah, very nicely invited all for 10th Monolith Summer Symposium next year. At the end, I want to thank the Sartorius BS operation to enabling this work. I want to say special thanks for my colleagues at PC4 Process Analytics department, also shown here on the picture. And nevertheless, all the colleagues, especially the PC2 department for process development of mRNA and pDNA processes. So thank you for your attention. Charlotte, I give you the word.
Thank you very much, Nejc. Great presentation. If you can stop sharing your screen now, that would be great. And we will move on to the Q&A.
Great.
And if you do have questions for Nejc, do send those in. And anything that we don't answer today, we will try to answer by email afterwards, or we will answer by email afterwards. So do keep those coming in. Brilliant. So first question I've got here for you, Nejc. Can you differentiate between full and empty LNPs using this analytical setup?
Yeah, this is quite good question. I will say that with this kind of configuration of the system, this is not possible because OH column, which binds the LNPs in the first step, cannot differentiate between empty and full LNPs. Probably some kind of other column modality should be used. For example, we make some experiments using ion exchanger columns for that. But yeah, in this kind of system configuration, this is not possible.
Okay, brilliant. And can you detect degradation product in LNP formulations?
Yeah. The nucleic acids are analyzed using reverse phase column, and the reverse phase column is known that it can separate that degradation products happening inside of LNP, for example, or degradation of free mRNA. So yes, it is possible, and yeah. You can use this kind of system to detect degradation inside of LNPs or outside of LNPs. Yeah.
Great, thank you. How big, how large can the nucleic acids be that are analyzed using this setup?
Can you repeat? I didn't understand the question.
With this analytical setup, how big can the nucleic acids be?
Probably there is no limit in the size of the nucleic acids analyzed on this kind of system configuration. We tried pDNAs and, for example, mRNAs up to 20K size, so probably even more. It will be just dependent on the concentration of acetonitrile you needed for elution of this kind of molecules. And one another thing, the pore size of the column could be a problem, but we can offer you also the columns with bigger pore sizes, and I think that there is no problem for analyzing over also the bigger nucleic acid at 20K.
Great. So sky's the limit there, really. To a point. So what would be a typical sample volume to work with?
So in all the chromatograms I showed today, the injection volume was 50 microliter, but this really depends on the sample concentration. If the sample has very low concentration, you will need to inject more. But LODs and LOQs of the method is quite low, so yeah, in the range of 10 nanograms, at least 10 nanograms of nucleic acid should be loaded on it, and this will be sufficient to successfully quantify and detect your nucleic acids. Yeah.
Okay, great. So at least 10 nanograms of nucleic acid. Brilliant. Yeah. So another question here. It's a little bit of a long question. Bear with me. So firstly, I want to say it was a great talk. So that's always nice. They wondered, are the LNPs destroyed for the analysis on the second column?
Yeah. We opened the LNPs, increasing the concentration of acetonitrile. Yeah, they're destroyed. We cannot quantify the nucleic acid inside of the particle if we don't destroy it. Yeah, we some kind of dissolve it, or we can say open it, and the payload is released and then can be separate and also detect with the detector. Yeah, we open the particles, yeah, on the second column.
Okay, brilliant. Hopefully that answers
but I will add also this: the particles are not destroyed eluting from the first column.
Right.
The particles stay the same. They are opened only on the second column. Yeah.
Okay, brilliant. Hopefully that cleared up the confusion for that viewer. If not, do send another question. Another viewer here asks, can you look at the payload?
So I don't really understand the payload. Yeah, we are looking the payload. We can analyze it, quantify it. We can separate it from other components of LNPs. Yeah, we can also detect some degradation if it is present inside of the LNP. So yeah, we can see the payload, really.
Brilliant. And is this method also applicable to other lipid nanoparticles? So things like liposomes, lipid nanoemulsions, lipoplexes, so on.
I think that it could be. We didn't really try it on so many different samples, but probably with optimization of the conditions for loading of these particles on the first column, and then optimization of the analysis of free and payload in the second column. Yeah, this method could be useful also for other LNP particles, how I say.
That's not something you've tested yet.
Not so extensive that I'd be really sure that this is possible. Yeah.
Good, excellent scientist answer. Are the LNPs heat-lysed to separate lipid from cargo, and does this affect the mRNA?
Can you repeat it, please?
Are the LNPs lysed by heat to separate the lipid from the cargo?
Not intentionally. Probably it could help to have a little bit elevated temperature for for the second column, but the really opening is happening because of the acetonitrile in the buffers. Yeah.
Yeah, and does that heating, if that heating does occur, would that affect the mRNA?
I think this separation is happened so quickly that also the elevated temperature does not influence on the mRNA integrity. But I could add the elevated temperature is used to because of the better behavior of mRNA on the reverse phase column. So we obtain nicer chromatograms and better separation also between different nucleic acids if the temperature of the SDVB column, so reverse phase column, is a little bit elevated. Yeah.
Great, thank you. And I think going back to the question about the size of LNPs, what size of LNPs have you tested?
We try different sizes of LNPs from, I don't know, from 80 nanometer to up to 200 nanometers. And yeah, we get quite consistent results if we compare it with other techniques used for determination of size of LNPs. Yeah, it doesn't really matter, I will say. It's just important that we can assure that nothing is happening with the particles when they are eluting from first OH column.
Okay, great. So in the region of 80 to 200 is what you tested.
Yeah, the pore size of the column is 6 micrometers. So I will say that that is a limit.
Yeah.
Yeah.
Brilliant, thank you. I think we've got time for maybe one more question. How do you use OH binding to separate LNPs from unloaded RNAs?
So yeah, this OH column is hydrophobic interaction column. So we load the LNPs with quite increase, or not so much, but with elevated salt concentration. So here we use citrate buffers. I will say around 20 millimolar of salt, and under these conditions, the LNPs are quite nicely binded on the disc column due to its hydrophobicity. But under this condition, the free nucleic acids, as the mRNA or pDNA, is not binded on the column. And one another thing, what I need to emphasize here is that we use basic pH, and this is quite important because if we go under the pKa point of the lipids, the lipid nanoparticles will be positively charged, and most probably the mRNA will stick on the surface of the particles. So also pH is here very, very important. And then the elution of particles happens just by lowering the concentration of salt, and in that way we can elute these particles from the column and analyze it. Yeah.
Great, I had another interesting question in. If any other solvent is used for the LNP degradation, does that interfere with the analysis?
For opening of the particles, or I don't know what.
It just says for LNP degradation in the question.
Yeah, in this case, we use acetonitrile because it's handy and is usually used also for reverse phase chromatography. But okay, we also perform some experiments with different surfactants and stuff like that for opening the particle, if this the question was. But it not works so well, especially because different surfactants are quite no good for reverse phase columns. and we stick with acetonitrile then.
Brilliant. Well, thank you very much for answering those questions, Nejc. I think that's all we've got time for today. Any questions that we didn't get to, as I said, we will reply by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And I think all that's left is to thank Nejc one more time for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.
Bye-bye.